Welcome, everyone. We are just a couple of minutes late. I want to thank you for coming, and I want to welcome Professor Luca Pinello, Department of Pathology at Harvard Division of Medical Sciences. I have been admiring a paper that was published just very recently called SIMBA. And I think Luca's approach to many problems in computational functional genomics will have tremendous impact, and I appreciate his coming to the conference and welcome him for his talk today.
Hi, everyone, and thanks for the nice introduction. It's such a great pleasure to be, and thanks for the organizer. And I really enjoy learning more about the bioconductor and interacting with you. So it's such a great community. So today I want to talk about functional genomics and try to understand gene regulation combining computational experimental approaches. And my background is in computer science, and I moved to ComBio. Try to think about these two questions. The first, can we understand the function of the genome and the functional elements that are important in disease or phenotypes? And also, how can we model cell-fate choices? We know that cells differentiate, and sometimes they essentially transform in states that are leading to disease. So these are very important questions, and today we'll focus more on the first one. I will present work related to understanding functional elements. But in the spirit of this community, I wanted to share that I love building tools, and there is such a joy, in creating tools for the community. And since I started the lab, we have been working on tools for single cell, CRISPR genome editing, transcription factor modeling. We have also this fun project where we play with single cell in virtual reality, and this is based on Google Cardboard. But we have been very active, and I'm very grateful for the lab that I have, and people really try to help the community. And so many of these are published, so I will not talk about. And thanks for the mention about SIMBA. This, we do a lot of single cell work, and we try to do maybe things that may model this data in a slightly different way. For example, in SIMBA, we use graph embedding techniques to create what we call regulatory maps, where you can embed cells, but as well also genes and other features. This is already published. And also last year we published this method based on single cell multiome that allows you to reconstruct gene regulatory network. But the nice thing is that we can create networks that are dynamic, that evolve along, for example, differentiation path. And then yesterday we learned about RNA velocity. We also work on this field, and we're trying to estimate uncertainty of this vector field. And we have a preprint, and also there is also a preprint about this paper made of SCDFQ. They use neural SDE to learn the most likely trajectories using a generative process. So, and today I want to work more to talk more about genome editing, given that we talk a lot about single cell and spatialomics. So I was thinking for this audience, it may be nice to get exposure also to these other experimental techniques. So, and there are a lot of work in progress. Today I will present this. It's called CRISPR Clear, and all of these are different ways to use genome editing to essentially study functional variants or element or cell states. So this Perturbo, for example, is a tool that we're developing to understand system trans effect of perturbation of an answer using as a readout single cell. And CRISPR-Beam, this is published recently, is a tool where you can perform base editing screening in bulk to assess effective variants or on coding sequences. And we develop a nice Bayesian framework to show like how can we improve these screens. So I don't have time to talk about all these methods, but I wanted to put some pointers, and also after the conference I would be happy to chat with you about these methods. So moving to CRISPR-Clear. So here, what we're trying to do is try to understand the relationship between genotype and phenotype, but we want to push the resolution of this exploration. We want to go up to nucleotide level resolution. So as I mentioned, one of my questions is try to understand the function of the genome. And I think, we are very lucky that, we have technologies to read, fully the genome. 20 years ago, we got the first draft, and, recently we got end-to-end references. But, one thing that, one quote I like from Eric Lander, and, it's an old one, we got the book, it's hard to read. I think it's still kind of true. And there are a lot of things that, we don't understand, especially in the non-coding part of the genome. That, is the majority. And, this non-coding part contain a lot of, functional elements. In fact, an indication of this is that many of the disease-associated variants are in this space. So it's very important to try to understand and dissect this space. So one, class of functional elements that we also learned yesterday are these enhancers. So these enhancers are regulatory regions that essentially mediate or promote or fine-tune expression of genes. And the way they work, the current understanding is that there are like transcription factors that recruit epigenetic modifiers. And usually these regions that are distal, if you think in a linear way, they will loop to a promoter, and this can promote gene expression. And we know that many of the variants are actually in these enhancers. So how can we understand this element? How can we find them, understand their function? As I mentioned, many variants, for example, that we can highlight from GWAS or QTL are in these elements. We can also use chromatin profiling, for example, chromatin accessibility or histone marks as profiled by ChIP-seq to find them. And also yesterday we learned about chromatin contact as a way to essentially decide or to assess which enhancer is controlling which gene. So, but one thing that is very important to assess function, in my opinion, is like to perturb this element. So we can essentially understand what they do to genes and phenotypes. So we are very lucky, now to have a lot of genome editing tools, and I'm sure you heard about CRISPR-Cas9. So CRISPR-Cas9 system, if you don't know, is a very nice, system that was engineered. So there is a Cas9 protein, and then there is like this piece of RNA called guide or guide RNA that essentially you can easily engineer. So you can design and synthesize whatever sequence you want, and this sequence would be used to target this system almost anywhere in the genome, assuming that you respect, this motif that is called PAM. So it's very programmable, it's very easy to use, and what happened after, this system engage and find the target, it will create double-strand breaks. So it will cleave the DNA, and of course the cell will try to repair, and this will give rise to short indels. And the nice thing is that we have also other tools that we can use. For example, people have disabled the cleavage activity, and so we have that Cas9 that doesn't cleave, but you can fuse with domains that essentially recruit epigenetic modifiers that can repress or activate the chromatin. So you can repress or create new enhancers. And more recently, we have these amazing editors. One is called base editor that use nickase enzyme and the aminase essentially can create substitutions. So you can go very precisely and modify the genome, or also prime editing, they use a reverse transcriptase to insert small, bit of DNA, and you can get very precise indel substitution. So we have a very nice toolbox to study the genome. So we can play different games. And one analogy that I like is that kind of play battleship against the genome. And imagine that there are like some region, you have some hypothesis, and the idea is like, depending on where you want to study, you can combine this perturbation and try to find maybe the critical spots. And today we will talk about a specific screen called tiling screen that is a little bit different than the usual genome-wide screen where you target all the genes in the genome. So the way it works, imagine that you have like this gene that you want to study regulatory elements. So what you can do, you can essentially design all the possible perturbations. So these are CRISPR perturbation, and you can potentially discover de novo these elements. But you can also use genetic perturbation, for example, CRISPR-Cas or base editor, to dissect now this element and uncovering critical nucleotide or transcription factor binding motif. And these screens are not novel. While I was a postdoc in Weisheng Yuan lab, I had the fortune to work with Stuart Torkey and Daniel Bauer, and we designed and executed one of the first non-coding tiling screen. And this was aimed at try to understand the enhancer of a gene BC11A that is a potent repressor for fetal hemoglobin. And what we found with this screen, there was a weak spot for this enhancer, and if by perturbing just with an individual guide this enhancer, you can essentially reactivate fetal hemoglobin. And fast forward, this was used as a potential way to cure sickle cell disease and beta thalassemia. There were clinical trials, and early last year they approved the first FDA drug based on CRISPR. So there are a lot of things that we can do with the screens, and there are a lot of other regulatory regions that we don't fully understand. So just to explain this new screen that I'm proposing, I need to give you a little bit more details about the usual way we run screen. So as I mentioned before, maybe you have some hypothesis, you want to study a gene or a phenotype, you tile with all the perturbation. So these are the perturbations that we can design. Then you lentiviral transduce the cell, and this is like graphical depiction of this CRISPR perturbation. So you introduce one per cell, and then you need to have some kind of phenotypic enrichment. Imagine you can sort your cell based on the one that express the gene and the one that don't express the gene. So then the game is like, now that you have two populations, you want to figure out which perturbation was important. So you can essentially sequence the cells and simply count the abundance. And if you do some kind of enrichment score, you can maybe find the region, given that these were designed in the genome. So we know the locations, we can create these 1D tracks. So we already, work on this, and we have a computational method that was specifically optimized for Cas9 and CRISPR-I that's starting from this raw count. There are a lot of steps. Try to essentially reconstruct the most likely signal that represent the functional region, and also we have some statistical tests to call the region that are significant. But, this was in 2018, so we've been thinking, can we improve this? And one, thing that I want to present today is this new way to do screen called CRISPR Clear. The first step of the screen is exactly the same, and here I try to, have side by side so you can see the differences. So you tile your region with all the perturbation. You have like some kind of sorting, imagine based on the level of a gene, and you extract the genomic DNA. So in the old school screen, as I mentioned before, we count, so we sequence the location in the genome where the guide is introduced, And then we count and we map. And here we make the assumption that the enrichment of these scores is proportional to the functional edits. But one key point is that we don't observe the perturbation. We don't observe the genetic modification that we do to the genome. And this is problematic because the different guide RNA, they have different editing efficiency. So you cannot recover like the true effect sites if you don't see what is happening at the perturbed sites. And this raises another problem. So we have this low resolution because again we don't observe the nucleotide. We just know that this perturbation, this guide was in the cell. In contrast, we reason that maybe the most obvious and maybe powerful way to do this is to sequence the region that we are modifying. So here, imagine you modify this region, you can sequence, you can design primers, and you will read out the alleles that we create after perturbation. So here we don't know anymore which guide, but we know what are the edits that we are creating in this region. And now comparing, as we did before, the allele in the high and low population, we can reason about what are the important allele associated with the phenotype and try to, given this collection of allele, to recover the important individual nucleotide score that may be important, and also transcription factor binding that may be modulated by this substitution. And this is move away from guide RNA enrichment. So we want to model functional edits. So to do this, you can throw different approaches. Right now we are using an approach called millipede, and that was developed by Martin Jankowiak. And it's a very fast Bayesian variable selection method. So the input, we can essentially encode our allele and have 0, 1 indicating which nucleotide we are modifying, and Y can be modeled in different ways. That in one formulation you can have the relative abundance or something proportional to the lawful change, and then given this matrix and this vector, we want to recover the beta that tells you for each nucleotide how much this nucleotide is increasing or decreasing your phenotype. And also we have this nice posterior inclusion probability to assess which nucleotide is important. So let me show you one example of this screen. So here we were focusing on studying CD19 expression in now 6 cells. So this is a nice model for leukemia, and then CD19 is a nice gene because it's one of the target for CAR T cell. So the idea was okay, let's try to understand regulatory element for this gene, given that this expression can have therapeutic implications. So what we did was try to find answers for this, and we find this region that is chromatin accessible, and some conservation. Also, the entire region is decorated by this active histone mark. So we delete this region, and then by measure mRNA, we see that single or double deletion clearly show a decrease. So we know that this is a true answer. And then we designed the screen on this region. So we took this region, we took 200 guide RNA, and here we use 2 editors. I apologize, these things have a very weird name, EvoCDA, NGG, AB. But what I want you to remember is that one create C to T edits, and the other one create A to G edit. And one is PAM-less, so we can just design all the guides. The other one will require this NGG motif, so it's less dense. And then in terms of the phenotype, we measure CD19 antibody fluorescence, given that is a cell surface marker. And then we can essentially sort in high and low based on the CD19, and then we run in parallel these 2 screens, the old school and the new one. And one thing you want to see when you do this phenotype, this is based on flow cytometry. So if you delete the enhancer, the full region, you want to see that CD19 fluorescence is shifting. And this is a nice check. And then when you run the screen, each cell is a cell that will get one perturbation. So you want to see that some cell will shift, and then you have a positive control, essentially targeting CD19, deleting the exon CD19, or negative control. So you want to make sure you have a good dynamic range, regardless of the computational strategy. So we ran CRISPR-Serv, and one thing that I mentioned before, it was not designed for base editing, but we tried anyway. And what we see is that if you zoom in, is that the results are quite inconclusive. So this is like the raw log-fold change, is that the convolved one, and this is one editor, this is the other editor. There is this region that seems interesting because there are 2 predicted binding sites, but essentially nothing is significant. And instead, if you look at CRISPR-Clear, the same region, we can build this very nice and dense map where the output is not region, but you have individual nucleotides. So you have the 2 screens. Of course, these are very small to see. So let me zoom in. And here, what you can see, we have to essentially create this visualization. We can hover, but there are some key part of this visualization. And just for simplicity, we have 2 screens. We in silico combine them, so we can look at one. So the way to read this map is you have 4 rows, 1 for each nucleotide. The circle denote the reference, where you have before modifying, and then also below we put the reference, and we color 1 letter if we can modify with the base editor. Not all the nucleotides can be modified with the base editors, right? So we have some limitation with current technologies. And then you see this square, color square, so they're colored based on the beta. So if it's red, means that we are decreasing CD19, and blue, we are increasing. And the sides of the square is proportional to the peak. So essentially, you have all the information in this map, and you can say, well, this is something important, this is something important, and you can essentially explore the full 300 base pair. Here I show you a region that is of interest. So what we did here was trying to take all this beta and visualize as a track where the height of the track is proportional to the beta. And with this, you can start to compare with transcription factor binding motif. So these are, if you're not familiar, tells, for each factor, what are the binding preferences. For example, in this nucleotide, you need to have an A. In this nucleotide, you need to have a G, and so on. Here, you can be more permissive. And what we see is that the model has high beta for nucleotides that disrupt the factor. So, for example, G is not tolerated by AKZF1, and so on. So the idea is that maybe CD19 expression is changing because this substitution are disrupting this transcription factor binding site. And another nice example, we find these 2 factors. So there are modifications that have no effect at all. For example, if you look this A to G, there is no letter here because the beta is 0, and PAX5 actually like to have a G here. So actually here we are potentially helping to retain the binding. So these are the kind of things we can start to do with the screens. And of course, we wanted to validate, and by ChIP-seq, we see that this factor indeed bind at this region, and by using individual perturbation that target this region, we see a nice shift for both this EvoCDA and AB8A screen. So confirming that this framework seems to work well, and we can learn a lot of information. So here, in summary, we have this new tool called CRISPR-Clear to investigate genotype to phenotype relationship. We can perturb and measure edits at endogenous loci. We can recover functional nucleotides at higher resolution. We can prioritize transcription factor sites and assess their nucleotide preferences. And this was a nice teamwork, with several people. These are some pictures of my lab, great, crew, and in particular Bashir and Zain and Justin that helped with this analysis. But this was a great collaboration with David Serrugia lab, with Sandra leading the experimental work, and Daniel Bauer Lab, and also Martin Jankowiak from the Broad Institute. And I still have some time, so I want to hold the question for the end, and I want to talk about another story. How much time do I have? Okay, perfect. So I just show you one way, we can learn the function of the genome by destroying. And then, more recently, with this idea, can we learn by creating, right? So what does it mean? So let me show you. So we have, this project called DNA diffusion, where instead of trying to understand regulatory elements by perturbing and destroying, here we want to create new regulatory elements. And if we really learn about the rule of the functional genome, these elements should work and should activate gene expression or open chromatin. So, and this is inspired by the wave of generative AI tools that have been developed in the past couple of years. I'm sure you use ChatGPT or Llama. We heard also talk, so you can easily create text. We can also create images, and we can just providing a simple prompt, we can create these funny images. And people have started to use generative AI also to create antibody and proteins. But for DNA, there's some work, but the idea was why don't we try to use some of these techniques and see if we can adapt them to our task. And one thing that you need to keep in mind is that if you want to create regulatory sequences, the genome is the same, almost the same for all the cells in our body, but they do very different things. So if you want to train this model, you should focus on regions that are important for gene regulation. As I mentioned before, we can potentially focus on distal elements like enhancers or promoters. And these are the switches that control if a cell will become a skin cell or a muscle cell and so on. So going back to the generative models, so the class of generative model we are using is called diffusion. And diffusion is a very simple idea, but so powerful. So the idea is that you want to learn to create points from a given data distribution. In this case, we have the distribution of the dogs. And the way you can learn that, you take a sample from this distribution and you gradually add noise until essentially you don't distinguish this from pure noise. But during this process, you can train a neural network that given a step, like let's say you're here, will learn to denoise, and go to the previous step. And if you repeat this multiple times, the hope is that from pure noise, you can denoise, like the original image. This is during training. But then during generation, you can create new noise, and you can apply the same procedure. And people have shown that you can actually create points that seems to come from the distributions. In this case, we can create dogs. And so these are very powerful model and very scalable. But one thing that a nice addition to this is that you can use what is called conditional diffusion. So you can essentially add some kind of guidance, in this case, a prompt. And this is called text-to-image diffusion. So you can essentially say, hey, I don't need a dog. I don't want a random animals. I want an astronaut riding a horse. So the model will try the best to do that. So we adopted this kind of technology, and here we want to create regulatory elements. So the idea is that imagine that you have a sequence in the genome that is important in this cell type, and here we want to switch this to another cell type. So the question is okay, which sequence do I need to create this switch? And why this is important, and I will show you some application, but before I want to show you the way we train this model. So we take data from a large set of chromatin accessible sites, and this is something that Wouter Mulleman curated. So in this case, I will show you one example with 3 cell types. We take cell type specific peaks, and then we extract the sequences. And given these sequences, essentially we apply diffusion that, in our formulation is based on a kind of a unit, if you're familiar with this architecture, where you take your sequence, you add the noise, you know the step of the noise and the cell type label, and then the model needs to predict the noise that you added, right? So you do this multiple times for multiple cell types, and then during generation, what you can do, you can reverse the process like I showed you before. So now you have a random sequence, a label, and you use your train unit in different steps, the noise in step, until it will converge to a sequence. So now you have a sequence that should be active in this cell type. So the reason why we think these models are interesting, there are like several applications. One is like context-dependent, regulation of gene expression, and this can be applicable in many contexts. So you can replace an answer. There are some AAV therapies, we learned yesterday, but you can also use this sequence to create biosensor that maybe will drive fluorescence in tumor cell, but not in normal cells. So, and I'm excited to hear other application maybe from you. But let me show you like what we did. So we trained this model, and then to check if it was working, we created 100,000 sequences for each of these 3 cell types. And then the question is like, okay, how do you know that these sequences are meaningful? Now you created things that are not in the genome. So the first thing that we did was to check transcription factor composition, and because we have well annotated transcription factor databases. We can also use oracles that can predict, for example, gene expression upon replacement of a distal region, or model like Luca-Pinello, they can predict chromatic accessibility, or model that recapitulate massively parallel reporter assays. So I will just show you some quick vignette. So for transcription factors, just one sequence that we create from scratch, and this was for FG2. And then one thing you can do, like you can compare the frequency of usage of this factor in the sequence that you create versus endogenous sequences. You see there is like good correlation, then there is this shift. And then, one cool thing is that these are the factors actually that are important for this liver cell type. So the model kind of learn, important factors. I try to create cell type specific and distinguish this sequence from the other 2 cell type, and also, use this sequence more abundantly than other cell type. Of course, you can formalize, and have a score. Here we use a Jensen-Shannon distance to show that, we have better concordance in the target cell type and, very high distance, in the other cell type, suggesting that the model actually learn to create, to place the correct transcription factors. So to show that, these sequences actually may have potential in opening the chromatin and reactivating gene expression, we use, these different oracles predictors, and I will show you just some quick result. So here you have the 3 cell types. Here we are creating sequences for FG2, so we want to see activation just in FG2. And as a reference, so these are the blue that we create, and the orange are the endogenous one. So we want to see that we are similar or better than the endogenous sequences, so we can discriminate. We don't have activity or low activity in the other cell type, and this is consistent for all the oracles that we created. Opening chromatin, activating expression, or this assay prediction. This is consistent also for all the other cell types. So the model seems to learn and discriminate and create sequences that work in the right context. Just to give you one concrete example of what you can do. So this is the GATA1 locus. This important gene in erythroid cells is a transcription factor, and there is this enhancer that has been CRISPR validated. We know that this enhancer is controlling the expression of GATA1. So these blue tracks are expression tracks, and these are the chromatin accessible tracks. You can see that there is a peak that is specific in KF62, but there is nothing else in the other one. So the game is like, okay, give me one sequence to flip this, and I want to have this now like open in FG2, and I want to see some activity in FG2. So we did this, and this is exactly what we see. So it's very nice that we can essentially flip the switch, and now we can start to see this GATA1 expressed. This is, of course, like in silico. So I want to show you one example, a real example in cells, because up to here you can be dubious, like, well, these are just predictions. So we focus on this gene AXIN2 that is important in leukemia. So there are like people that have, leukemia have low expression, they have a poor prognosis, but some people are lucky that they have a variant, they have a protective variant that essentially change dramatically the chromatin, create enhancers, and start to reactivate AXIN2, and they have a better survival. They have like 10 years. And the idea was like, can we create a sequence that is even better than this protective variant, so we can potentially envision a therapy where we can replace this region and activate AXIN2. So we had a very nice collaboration with Judith from the Planck lab at PFL. So we essentially tried to do this, replacing in cells, and we use this GM11278 that is a very similar cell type, and we selected different sequences spanning a dynamic range of intensity and specificity, and also including sequences not designed for this cell type. And she has a very cool assay, so you can essentially replace at the endogenous site the enhancer, and we use 100 replaced sequences, and then measure the log-fold change between the DNA that you integrate and the expression. And if you do this log-fold change, it's a proxy for the activity of the DNA that you introduce, correcting for how many integration you have. So it's kind of NP array, but endogenous readout. And so these are the results. So this is the x-axis, the AXIN2 reactivation. So these are positive control, and then the black line is like the wild-type sequence. So what you expect, like if you don't have any variant. This red line is the protective variant, and then we have the groups. So as I mentioned, we sample from a range of activity and intensity, and this is well recapitulated here. And we can clearly see that we can select and create sequences that exceed our goal, that would be the protective variant. Importantly, sequences designed for other cell type or negative control don't have any effect. So we were very happy to see this. And then we asked, like, now that we sampled this range, what are the most predictive features of things that will reactivate? To our surprise, DNase was not show a good correlation, but one thing that showed a very good correlation was CAGE. So this is like, this enhancer activity. And, this was very, maybe not super surprised. Other people have described this, but now we have a way to potentially select sequences. So in conclusion, I show you DNA diffusion, that is a new way to create synthetic regulatory elements that are cell type specific. I show you like our in silico evaluation using different oracles to predict their function. Important, I show you some example in real cells where we can reactivate AXIN2 expression, and this can be a potential future therapy for leukemia. So with this, I would like to acknowledge people in my lab, in particular Luca, Simon, and Zain, that worked really hard together with a nice online community, Open by ML, and several key people that help with the experimental design, data collection, and also analysis. And with this, I will stop here. I will take questions. Thank you.